Hello, everyone. Before we begin today with chapter 12, I want to give you a little review of things you should be familiar with before we get started on biosignaling. As we continue to the second half of the semester, we are moving from learning just the structure of our biological molecules to their function, as well as the chemical mechanisms that allow our bodies to function. What did we learn so far? We learned the structures of several of our macromolecules that we'll be utilizing in this chapter. First is proteins. So you know the structure of proteins. They are heteropolymers of 20 amino acids, such as tyrosine. And today we'll be looking at the structure of tyrosine and how it acts catalytically, as well as our other amino acids that have polar groups, as well as alcohols as their functional group. So I suggest you become very familiar with the structures of all 20 amino acids. Maybe learn a song. I put a link to a pretty bad song of the 20 amino acids, but you can learn any song that you want on YouTube to try to make sure you remember the structures of the 20 amino acids. The second macromolecule that we learned the structure of was carbohydrates. Carbohydrates can be homopolymers or heteropolymers of saccharides. You know the structures of many of those saccharides, such as glucose and galactose and lactose. Make sure you remember the structures of all the carbohydrates that you were told to memorize. Make sure you're familiar with table 7.2. The structures and roles of the polysaccharides, and also make sure you're familiar with for the test with figure 7.22 and be able to distinguish between the 4 glycosaminoglycans. We also learn about the structures of lipids. We have storage lipids that have fatty acids called triglycerols. We have membrane lipids, so we're going to be talking about biosignaling, which includes the lipids that are in our cell membranes, the phospholipids, the sphingolipids, the glycolipids. So make sure you're familiar with the structures of those, as well as some of our lipids that do not have fatty acids, such as our steroid hormones that look a lot like cholesterol. Some are precursors to other molecules that we'll be talking about. And especially make sure you're familiar with signaling molecules that are lipids, such as arachidonic acid. The last macromolecule that we learned the structures of were nucleic acids, RNA, DNA. Make sure you're very familiar with the structures of nucleotides. That's nucleotide with a T. So nucleoside is a nitrogenous base plus a ribose sugar. A nucleotide is a nitrogenous base plus the ribose sugar plus a phosphate. So when I say nucleotide, it can be ATP, adenosine triphosphate, or AMP, adenosine monophosphate. Those are both nucleotides. So we can have ATP, GTP, CTP, things like that. So make sure you be able to quickly recognize what a nucleotide is. As we begin today, there are a few terms you should be familiar with so you won't get caught up in the words that I'll mention today. First of all, a receptor. You probably know what a receptor is. A receptor is a protein that sits on the surface of a cell membrane and binds to a signaling molecule. So just make sure you understand that it is a protein and it binds to a signaling molecule. The next is a kinase, so it ends with ase, so we know that it is an enzyme. A kinase is responsible for transferring a phosphate group from one molecule to another molecule. Usually it's responsible for taking a phosphate from ATP or GTP and transferring it somewhere else or removing it. So a kinase is a molecule or an enzyme responsible for transferring a phosphate group. You'll see other ACEs such as adenylyl cyclase, and it's a cyclase, so we know it's responsible for catalyzing the cyclization. This one we'll talk about today is responsible for cyclizing and a group that has an adenosine on it. We'll also be working with lipases, and lipases are responsible for cleaving lipids. So just make sure you understand what an ACE is. An ACE is an enzyme that's catalyzing some reaction, and a kinase is responsible for transferring a phosphate group. Chapter 12, Biosignaling. The first objective for Chapter 12 is to learn the general features of signal transduction. That means in what general ways do cells take stimulants or information from the outside of the cell and turn that into a chemical response or a chemical change inside of the cell. We will then learn the structure and function and the mechanism of G Protein Coupled Receptors. Next, the structure, the function, and the mechanism of another example of biosignaling, enzyme-linked receptors. And our last objective is to learn the structure and the mechanism of function of gated ion channels. What is the biological role of signal transduction? Well, cells receive signals from the environment beyond the plasma membrane. That's information. So right now, of course, you are at home because of the coronavirus. That's an antigen. That antigen enters your body, and your cell membrane receives that as a signal. And hopefully, your cell is eliciting a response, such as produce antibodies that can fight this antigen. Another signal from outside of the plasma membrane might be a hormone, such as a steroid hormone, like testosterone or estrogen or something like that. It is outside of the cell. It binds to a receptor, and that receptor sends a signal inside of the cell to produce more of some gene or some enzyme or some protein inside of the cell. The next one is light. We actually talked about a signaling hormone that responds to light, like beta carotene. So beta carotene is just vitamin A. Your body takes in vitamin A, it cleaves it to produce a lipid called cis-retinol. That cis-retinol is changed into all trans-retinol by a photochemical reaction with light. That is a signaling molecule that tells your brain what you're actually seeing. Lastly is touch. So if you were to just pinch yourself, say, ow, well, that is a signaling molecule being sent to your brain to tell you that you feel touch or you feel pain. We talked about arachidonic acid, which is in your cell membrane. It gets cleaved, and you pinch yourself, it forms arachidonic acid. That arachidonic acid can turn into prostaglandins, which is a signaling molecule that gets so quickly sent to your brain to tell your brain to respond in a particular way that you are in pain. These signals cause change in the cell's composition and function. That's a chemical change. It can elicit changes in amounts of enzymes or metabolites. It can elicit changes in permeabilities of membranes, differentiation and antibody production, or growth in size or strength. So first we receive information outside of the cell. It binds to receptor on the cell membrane, and then we'll see that receptor makes a conformational change that causes a chemical change inside of the cell. Our first objective is to learn some features of signal transduction. Your book outlines about 6 features of signal transduction. You should be familiar with these 4. The first feature of signal transduction is specificity. This is identical to what you already learned in protein ligand binding. So a signaling molecule fits into the binding site of a complementary receptor. It's complementary in its intermolecular forces such as hydrogen bonding or ionic interactions, whereas other signaling molecules do not fit. The second general feature of signaling transduction is particular to signal transduction, and that's amplification. So we'll see a signaling molecule would bind to a receptor. It will cause a conformational change, which would activate an enzyme. That enzyme might create a second messenger. So we'll see that come up over and over again. A second messenger is created. That enzyme could create many, many second messengers. And those second messengers can go on to activate many other enzymes. And then other enzymes will be activated. And that just completes a cascade of events where one signaling molecule can activate many, many enzymes upon activation. The third feature you should be familiar with is desensitization and adaptation. So once we have that signaling molecule binding to a receptor, we're going to cause a response in the cell. But the cell has to have a way to turn off that signal so we can stop the response, and that's desensitization. The last feature you should be familiar with is integration, and that means the cell could have 2 receptors that are giving opposite responses. So basically the cell will come to an equilibrium. Well, it can have the right response and the right concentration in response to 2 different signaling molecules. Make sure you're familiar with these 4 features of signal transduction. On the bottom, I have a link to a pretty good YouTube video that explains it with animation in detail and a lot of detail that you should know. But if you want to take a look at it, I think it's a pretty good video. There are several signal transduction mechanisms that are very well studied. You can see there are 6 of these on this slide. We are going to focus on just 3 of those that I mentioned in the objective. There's G coupled protein receptors. We'll learn about receptor tyrosine kinases as well as gated ion channels. The first one we'll learn about is G coupled protein receptors. This receptor sits on the cell membrane. It binds to its complementary ligand. That receptor is associated with a G protein. That G protein gets activated, and it activates another enzyme, and that enzyme produces a second messenger. So we'll learn the details of that mechanism. The second very well studied system is the receptor tyrosine kinase, where the ligand binding activates a tyrosine kinase. It gets autophosphorylated. That autophosphorylation leads to a molecule that can go to the nucleus of the cell and turn on gene translation and transcription. We'll learn about that in the next lecture. The last objective and the last system we'll learn about is gated ion channels, where our channels open or close in response to concentration of signal, ligand, or membrane potential. So the ligand binds, and that causes a conformational change that allows ions to go in or out of the cell. So again, we are going to become familiar with just 3 of these signal transduction systems: the G protein coupled receptor, the receptor tyrosine kinase, and the gated ion channels. G protein signaling is the first mechanism you should learn. The G protein signaling involves a G protein coupled receptor, a G protein, a GTP, guanosine triphosphate, and a target enzyme. The external signal binds to the G coupled protein receptor, that is an alpha helical integral membrane protein. We see that over here. Let me get my mouse. This red one here is the hepta alpha helical membrane receptor. Here is called the beta adrenergic receptor because it's a receptor that binds to adrenaline or epinephrine. It's called beta because there are several of these. There's alpha 1, alpha 2, beta 1, beta 2, and we're going to be studying the beta 2 adrenergic receptor. It's just a synonym for the G protein coupled receptor. So it spans the cell membrane, and of course the ligand binding site or the epinephrine binding site is outside the cell. It spans into the inside of the cell, and it is associated with a G protein. And it's called a G protein because it binds to a GTP. That G protein is heterotrimeric, and it has an alpha subunit, a beta subunit, and a gamma subunit. So we have our receptor on the outside of the cell, which spans through the whole cell membrane. It's associated with a G protein, called a G protein because it binds GTP. And that G protein has 3 subunits: an alpha subunit, a beta subunit, and a gamma subunit. And this G protein is only active when it's bound to a GTP. When it's inactive, it's bound to a GDP. This G protein gets activated by binding of the beta adrenergic receptor, and it can actually dissociate, inactivate its target enzyme, which is not shown here. The signaling molecule that activates the beta adrenergic receptor or the G coupled protein receptor is adrenaline. We call it adrenaline or we call it epinephrine. Those are the same thing. It's the fight or flight hormone. So as you see a bear or you see a child stuck under a car, epinephrine starts to be produced in your body and it binds to the beta adrenergic receptor. We see over here to the right the structure of epinephrine. If you look at it, it kind of looks like an amino acid. Hopefully you see that. It's actually just a derivative of tyrosine. Epinephrine, we see, has a KD of 5 micromolar, so it binds pretty well to the beta adrenergic receptor. We can understand the structure of epinephrine and make analogs to epinephrine that can be helpful medically. So underneath the structure of epinephrine, we have the structure of an agonist, isoproterenol. It's an agonist because it binds to the same receptor as epinephrine and elicits the same response. Its KD is even smaller, which means it's a better binder, which means it elicits more of a response. So this agonist is used to get more of a response than epinephrine. Usually you'll see it as a drug that helps to raise the heart rate. Because when you think of epinephrine, you think, oh, I see a bear and my heart starts to raise. So if we have an agonist that's even better than epinephrine, it can start to raise a slow heart rate. We have another analog of epinephrine, propranolol. This is an antagonist, however. And an antagonist binds to the same receptor, but it doesn't elicit a response. So in that case, it's an inhibitor of epinephrine because it's binding in the same binding site. So if we look at the KD of this antagonist, it's even lower than epinephrine. So therefore, we can completely bind to the epinephrine binding site and stop epinephrine from doing the function that it does. And so it would be used medically to actually stop high blood pressure or slow down someone's heart rate. So we can utilize the function of our signaling molecule, and medically we can use those to either get more of a response or less of a response. The epinephrine hormone is made in the adrenal glands. It's a pair of organs at the top of the kidneys. Again, it mediates a stress response. The binding to receptors in the muscle or liver cells induces the breakdown of glycogen. So it's a signaling molecule that's very specific to certain sites of cells. And we know that epinephrine is used to give us energy so that we can run really fast from a bear or a dog or do something like that. So it would bind to muscle cells so it can give our muscles energy. And it will bind to liver cells, and we know that liver is where we store our glycogen. So what is glycogen? It's a homopolymer of glucose. So if we have the breakdown of glycogen, then we're producing more glucose. We produce more glucose, we produce more energy. It also binds to receptors in fat cells because fat is another source of energy for us. It also binds to receptors in heart cells to increase our heart rate. So the G Protein Coupled Receptor, or the Adrenergic receptor, binds specifically to epinephrine to give us energy by breaking down glycogen in our liver to produce glucose. It binds to our muscles to give energy to our muscles, or to our fat cells to break down lipids to give us energy. There are many G proteins, but the composition of those G proteins is tissue specific. The beta adrenergic receptor signaling pathway that's in our liver, in our muscles, in our fat cells, which I've just mentioned, is the best understood. So you're responsible for learning the steps and the mechanism of the beta adrenergic receptor pathway. So let's go over this pathway. Step 1, the epinephrine binds to the specific receptor. So that makes sense. We have our signaling molecule, which is epinephrine. It binds to a specific receptor. This causes a conformational change in the receptor that also causes a conformational change in the associated G protein. Remember that the G protein, it's the one in green, has 3 subunits: an alpha subunit, beta subunit, and a gamma subunit. The alpha subunit is where the GDP nucleotide binds. Once it has a conformational change in the G protein, that causes the GDP to dissociate from the alpha subunit, and a GTP then binds to the alpha subunit. So we don't have dephosphorylation or phosphorylation of the GDP. We have the GDP, after a conformational change, actually exits the alpha subunit, and a GTP enters the alpha subunit. So now that the GTP, guanosine triphosphate, is bound to the alpha subunit of the G protein, it's now activated, and actually it just moves along or dissociates from the receptor. So it leaves the beta and gamma subunit and moves along the cell membrane and finds adenylyl cyclase. So we see it's the cyclase, so it's going to cyclize something with the adenine in it. So this now activated alpha subunit of the G protein is going to associate with adenylyl cyclase. This then causes a conformational change in the enzyme adenylyl cyclase. This enzyme is going to bind to and act on an ATP. So there are many ATPs already in the cell. Once this adenylyl cyclase is activated, it can catalyze the reaction that turns an ATP into a CAMP molecule. Adenylyl cyclase gets activated, it turns an ATP into a CAMP. That's cyclic adenosine monophosphate. So a very similar molecule, but we see that the ATP has now lost two phosphate groups because now it's AMP, and that phosphate group has become cyclized. CAMP, you'll hear over and over again, is a second messenger. Now, as we saw in our general features, the second messenger has the ability to activate many other things. So now that we have created CAMP, that CAMP has the ability to allosterically bind to pKa. This is step 5. Once we've created CAMP, CAMP activates pKa. What is pKa? It's protein kinase A. Protein kinase A is activated, and it does what kinases do. Kinases phosphorylate other things. So it phosphorylates in step 6 many other proteins. When it phosphorylates other proteins, it turns them on or activates them. So CAMP activates protein kinase A. Protein kinase A goes around the cell and activates many other proteins. Some of those proteins might be proteins that break down lipids or break down glycogen. And when you break down lipids or glycogen, then you in turn produce molecules that give us energy. In step 7, that CAMP can be turned off by being degraded into AMP, which is not a second messenger. And so AMP is created from CAMP, and now we no longer have that second messenger, and the system is shut down. So we need a way to desensitize everything and turn off the response. And that happens by another enzyme that converts CAMP into AMP. So I want to go over the mechanism one more time. The first step is the epinephrine binds the receptor. The receptor has a conformational change. That conformational change causes a conformational change in its associated protein, the G protein. The G protein has 3 subunits. The alpha subunit trades its GDP for GTP. The GTP-bound G protein moves along the cell membrane. It finds adenylyl cyclase. It activates adenylyl cyclase. Adenylyl cyclase turns ATP into CAMP. The CAMP is a second messenger that activates pKa. pKa phosphorylates other enzymes, such as those that are responsible for breaking down glycogen. Once glycogen is broken down, it can actually form glucose molecules that can be converted into energy. There's a phosphodiesterase that is involved in turning CAMP into AMP, which shuts down the system, so we will no longer have that response. The real player in the G protein mechanism is the CAMP molecule. CAMP is a second messenger. It activates the CAMP-dependent protein kinase A, or pKa, protein kinase A. The pKa activation leads to activation of enzymes that produce glucose from glycogen. It also opens certain ion channels, and it performs many other regulatory functions. Let's just look at the structures of the molecules that are active upon in this mechanism. So here we have the structure of ATP, which you should be familiar with and you should memorize. We have our adenine, we have our ribose sugar, and 3 phosphate groups. So this is ATP. We see that when adenylyl cyclase gets activated, it catalyzes the conversion of ATP to CAMP. So CAMP is adenosine 3'5' cyclic monophosphate. So we've gone from triphosphate to monophosphate. So we lost 2 phosphate groups. And then we've cyclized the phosphate group, and so now it's 5', 3' phosphate. So usually our phosphate is usually connected to the 5' end of the ribose sugar. Now it's cyclized and connected to the 3' end of our ribose sugar as well. So that CAMP molecule, it goes along and it starts to activate protein kinase A. But that signaling molecule binds allosterically to protein kinase A, so it can activate many protein kinase A enzymes. So we want a way to intrinsically shut down CAMP molecules so we can stop that response. And so we have another enzyme that gets involved. It's called cyclic nucleotide phosphodiesterase. So that as many CAMP molecules are created, they can be broken down to AMP, adenosine monophosphate. And so those CAMP molecule concentrations will get lowered, and we can stop phosphorylating the enzymes that will produce energy. One of the general features of signal transduction is amplification, as we mentioned before. So if we look at the right, one epinephrine molecule can bind to one epinephrine receptor complex. It's associated here to one G protein, but it can associate to many G proteins, one after another, as they move along and find an adenylyl cyclase. And if one is activated, it can activate many adenylyl cyclases. So one epinephrine can activate as many as 10 G proteins, and they can activate 10 adenylyl cyclases. Every active adenylyl cyclase enzyme can make several CAMP molecules, thus activating several pKa enzymes. These activate thousands of glycogen-degrading enzymes in the liver tissue. At the end, tens of thousands of glucose molecules are released into the bloodstream. If tens of thousands of glucose molecules are in the bloodstream, we can produce millions of ATP molecules. So we can see amplification by just one signal molecule. So let's follow along on the right of the slide. We have one epinephrine molecule. It binds to one receptor. It activates many G proteins because the G proteins can associate and be activated by one receptor. So then maybe we have 10 G proteins that are activated. They can go along and activate 10 adenylyl cyclases. Each adenylyl cyclase can activate 10, 20 ATP molecules and turn them into CAMP molecules. So now you've created 200 CAMP molecules from just one epinephrine. Those 200 CAMP molecules can activate 200, 100, many pKa enzymes. When you activate pKa enzymes, they go along and they phosphorylate many other enzymes, such as those responsible for degrading glycogen. Once you have done that, you can create thousands of glucose molecules from many glycogen breaking down enzymes. And once you have thousands or tens of thousands of glucose molecules, we'll see that those glucose molecules can give us dozens of ATP molecules. Another feature of signal transduction should be termination of the signal. There are 4 methods for stopping the epinephrine signal, and they are used simultaneously. Because once we turn on the signal, we need a way to stop the signal and stop our body from producing ATP or adrenaline. One method is an intrinsic GTPase hydrolysis. So intrinsically, our GTP can be converted by GDP from the G protein, causing the subunit to deactivate. So remember that the G protein is inactive when it's bound to GDP and active when it's bound to GTP. So once the GTP is associated with the G protein, intrinsically it eventually dephosphorylates to GDP and becomes inactive. The second method is that a series of additional enzymes bind to the G Protein Coupled Receptor, inhibiting activity and mark a receptor for sequestration in the inner membrane. This is called desensitization. So we'll learn one of the mechanisms of desensitization where we would inhibit the activity of the receptor and pull it into the inner membrane. The third method for stopping the epinephrine signal is that the epinephrine concentration drops below the KD. So once the fearful signal is lost, your body stops producing so much epinephrine, and the concentration of epinephrine drops below the KD, and so we get less binding to the receptor. We just saw the fourth way of stopping the epinephrine signal is that the CAMP molecule, the second messenger, is degraded by the enzyme cyclic nucleotide phosphodiesterase. So make sure you are familiar with these 4 methods of stopping the epinephrine signal. We will look in further detail to method 1 as well as method 2. Let's look at self-inactivation and G protein signaling. Epinephrine is only meant to be a short-acting signal. The organism must stop glucose synthesis if there is no more need to fight or flee. Let's look at the mechanism of intrinsic dephosphorylation. So step 1 says that the GS, or the stimulatory G protein with the GDP bound, is turned off. So usually, if there's no reason to fight or flee, there's a G protein receptor. It's coupled to a G protein, and it has an inactivated G protein because GDP is bound. Then, when we bind to a epinephrine, the GDP is exchanged with a GTP. That GTP bound G protein finds the adenylyl cyclase, and we have the activation that we saw before. Eventually, that GTP will get dephosphorylated to GDP. It will lose that phosphate group. Well, now it's inactive. It goes back and finds the beta and the alpha subunit, the beta and the gamma subunit, and associates back with the receptor in its inactive form. So the G protein eventually loses the GTP by dephosphorylating it to GDP, and then it can go back and find the beta and the gamma subunits and associate to the receptor in its inactive form. Another feature of signal transduction is desensitization. Desensitization causes a dampening of the signal even while the epinephrine is bound to the receptor. So we need a way to turn off this signal even while epinephrine is bound. Let's look at the mechanism of desensitization in our epinephrine receptor complex. So first we have the epinephrine binds to the receptor. We already know once it binds to the receptor, the G protein, the alpha subunit of the G protein, dissociates, and it goes along and it activates an adenylyl cyclase. Well, also the beta and the gamma subunits dissociate from the receptor. They move along the cell membrane a little bit and recruit a bark protein. A bark protein is a beta adrenergic receptor kinase. So they recruit this kinase, bark, to the receptor. The kinase does what kinases do, and it phosphorylates the carboxyl terminus of the receptor. Once the receptor is phosphorylated, it binds to another protein called BAR. Now this BAR protein is an inactivator. It marks the receptor to be sequestered into the cell. If the cell is no longer on the cell membrane, it can no longer accept epinephrine and no longer cause that cascade of events. It causes adrenaline to work. So in summary, we have the receptor that gets phosphorylated by the bark kinase. It then recruits the BAR inactivator and gets sequestered into the cell. The last topic for today is to look at a second broad class of G protein coupled receptors that activate phospholipase. So we saw before that the G protein coupled receptor, or the beta adrenergic receptor, eventually activated adenylyl cyclase. We'll see that this protein receptor activates phospholipase C, and it employs different second messengers than the CAMP we saw before. So we can go right into the mechanism. The first step is that the hormone, whatever hormone it is, now it's not epinephrine, it binds to the G protein coupled receptor. That receptor again is associated with a G protein. Now that G protein becomes active, by exchanging the GDP with the GTP. That activated alpha subunit of the G protein associates with phospholipase C. So before it activated adenylyl cyclase, and this mechanism is activating phospholipase. What do lipases do? Well, lipases break down lipids. So it's going to break down a very near phospholipid because it's a phospholipase. So we know that phospholipids are contained in our cell membrane. So it's going to break down this phospholipid called PIP2 into two different pieces. One is diacylglycerol, so we just recognize that the backbone of the phospholipid is glycerol, and the inositol 1,4,5 triphosphate, or IP3. So inositol is just a sugar, so it's a phospholipid that has a sugar on it that has a phosphate group in the 1, 4th, and 5th carbon. That IP3 is going to be our second messenger. That second messenger is going to bind to a calcium channel within the cell. That calcium channel is going to be opened up and it's going to release a calcium. That calcium is going to activate a protein kinase C. So the release of calcium is going to activate protein kinase C as well as the diacylglycerol that's already produced from the lipase. What happens when you activate the kinase? They phosphorylate. So we're going to see the phosphorylation of many other target enzymes, and they're going to activate other pathways that are going to cause a response in the cell. So this is a very, very similar mechanism that we saw before. It utilizes not a camp second messenger, but a group of second messengers that are created by activating phospholipase C. It's going to convert the phosphatidylinositol 4,5 bisphosphate, just a phospholipid that has a sugar on it called inositol with two phosphate groups, into two second messengers. We'll call those DAG and IP3. The IP3 is going to go along and activate a gated calcium 2+ channel, and then the calcium in cooperation with the DAG is going to activate a kinase. The protein kinase C is going to do what kinases do. They're going to phosphorylate many other target enzymes, and those target enzymes are going to cause a reaction and response in the cell to give whatever response is elicited by the hormone. Alright, I hope you learned a lot. My daughter was with me in the beginning. She fell asleep. I hope I didn't put you to sleep. And make sure you read your book, follow along, and you should do just fine. See you next time.